Everyone, my name is Gene Cismero, and I'm very excited to help out here as I'm taking over as chair. And it's my pleasure to introduce the first speaker in my time here. So today we have Jeremy Colmel, and what Jeremy is going to speak to us about today is his software, which is called FluoroMatch, and the title of his talk is Uncommonly Screened PFAS Are Everywhere, Answering the Need for Non-Targeted PFAS Analysis with FluoroMatch. And to give you guys some background, Jeremy is currently the CEO of Innovative Omics, a company involved in training labs in non-targeted lipidomics and PFAS workflows. He's also a researcher at Yale as part of the Crystal Polity Laboratory. He focuses on developing data processing workflows and software specifically for compound identification using high-res mass spec. He's most excited about environmental applications related to sustainability and protection of land and water. Jeremy's written over 50 publications in the last 5 years on related topics. He's led teams to develop 10 different software, including both vendor software, you may know Lipid Annotator from Agilent, and open source solutions. He's excited to continue expanding the frontier of compound identification using mass spectrometry. So with that, Jeremy, please take over and present away. Thank you so much.
All right, thank you for that introduction. So I think most of your background is in this group is biopharma, is that correct? Or just get an idea?
For the most part, I think, yeah.
Okay.
Yeah, it looks to be mostly that, yep.
All right, so I'll try to cater to that a little bit, but PFAS are interesting structures. I think many of you might have heard of this group of compounds. It's been getting a lot of attention recently, although for many decades it's been in the environment and been used. And the really interesting thing is the carbon-fluorine bond. It's the strongest bond in nature. Fluorine is the most electronegative. And so, or not found in nature, but now found as we create it. And that gives it unique properties. So if you look in the household, for example, it neither looks like a CH group, so it's neither lipophilic or fat soluble. Neither does it look polar, very polar, so it's not water soluble. And so it can't be destroyed very easily with that strong bond. So it has very good water repellent and oil repellent properties, so non-stick properties, which means that it's used in couches and chairs and rugs, and bed sheets, food wrappers, et cetera, cooking non-stick pans. So has really great
Tuesday and Wednesday is the wind settles.
And then also, because of these properties, it can be used in a lot of different types of fluidics, things that are used between metal parts and not be degraded for lubricant, things like that go under high heats. So it has a lot of firefighting foams. Because of this, and to have these unique uses, of course, there's going to be many unique structures. I think up to now, I would say there's 9,000. It changes every month. They add new ones to the EPA's database of PFAS, run by Anthony Williams. So it keeps growing, and there's a lot out there. Health-wise, they look a lot like fatty acids. I mean, of course, this carbon-fluorine bond is going to be a little shorter. Tighter is going to have different properties, but they do affect a lot of these lipid pathways, given the fact that they look a lot like fatty acids and affect some of the same signaling pathways, and may bind to some of the same receptors, so in the PPAR pathway, et cetera. So, for example, they've been shown to be likely obesogens, so leading to obesity. They affect adipocyte function, increase fat accumulation, exacerbate hepatic steatosis, and they affect a range of other systems: the endocrine system, immune system, reproductive system, neuronal system. And their health effects have been known for quite a while now. In 1984, kind of the start of this timeline, so 40 years ago, DuPont detected PFAS in the tap water in Little Hocking, Ohio. They didn't alert anyone about it, but they were dumping. There was dumping going on in Ohio. There was dumping going on in Virginia, different areas, right into the water systems there. In 1987, so this is kind of like a hot period of time, 3M started looking into animal studies and finding tumors with those with PFOA, which is that one that looks like fatty acid carbons. And then from 1989 to 1997, there was 3 different studies by 3M and one and some by DuPont as well, finding elevated cancer rates among PFAS workers. I believe thyroid cancer was one of them. And then in 1999, 3M scientists described PFAS as the most insidious pollutant since PCB. 2000, 3M PFAS and PFOA voluntary phase out with the EPA, these chemicals, which is a very important time and shifts kind of those that we find in the environment, which I'll show you soon. By 2012, this had started before 2000, but by 2012, the C8 panel, which was, there's some movies, Dark Waters and Devil We Know, which you might want to watch on the subject, very interesting kind of recreations of what happened. But the C8 panel of scientists working with these activists and people in the area linked PFOA and testicular and kidney cancer with 32,000 participants from these contaminated areas and workers. And so this was a large epidemiological study with quite significant results. So here's some of those movies, some mechanisms for which it might be carcinogenic. It induces oxidative stress, that's been shown, immunosuppressive, and modulates receptor-mediated effects. Suggested evidence shows that it can induce epigenetic alterations and influence cell proliferation. And then here are some of those relative risk factors gotten from the studies. To continue that timeline, in 2017, DuPont paid $670 million to settle the C8 lawsuits. And then Chemours is kind of like an offshoot of DuPont so that they can take the brunt of it. DuPont won't be sued. And yet they can still profit off these things. And so in 2021, DuPont, Chemours, and Corteva came to a $4 billion settlement. So obviously these kind of settlements, and I think that money's set aside for different people affected by PFAS, are not easy to win against these big companies. And so that, I think, is a testament to the risk of these chemicals in the environment. Now, from a analytical standpoint, if we look from 2000 down to 2016, and this trend continues on to today, we can see that PFAS and PFOA are starting to decrease. That yellow is PFAS. So some of these common perfluorinated chemicals that were widely used are decreasing. So this is amazing. This is great industrial phase out, working with other people, and then having activists also pushing this forward, decrease in PFAS on scientists. What this doesn't show for is all the other PFAS chemicals. So again, very important industrial applications. They're going to find alternatives, and these alternatives are mostly carbon fluorinated based compounds. And so what we see here on the left is that it starts to increase of this total PFAS, in this case indicates targeted. So known PFAS, most of these studies looking at 20, 30 compounds, start increasing up to 2001. And then after that voluntary phase out, you can see a rapid drop in the amount, sorry, this is in human serum in Sweden, drop in the amount in human serum. So amazing, awesome results. What you do see, though, is that the extractable organic fluorine in this middle plot does not have a trend towards decreasing. Although you can see in before 2000, there are some very high studies, but overall it stayed relatively similar in these populations across a number of studies. And so what that means is that the percent of those fluorinated, carbon fluorinated compounds out in the environment that consists of things that we know, or at least routinely screen is decreasing over time, and the percent of unknowns is increasing. And this has been shown in different, so that's in serum. This is showing in many European countries. It's also been shown in America, but this is in sewage. And you can see that the gray and kind of slash green there are unknown fluorinated compounds. And you can see that they dominate in many of these studies, many of these areas in terms of sewage. And so we really have a large number of these chemicals that we're not detecting at all with health impacts, and that are in human serum. So big study question, a lot of interest in PFAS, not as much focus at this time on the unknowns. And so this is just taking home that that's important. So the question, what are these PFAS? How much, how toxic? And the big thing we need for this, non-targeted mass spectrometry works wonderfully to detect all these unknowns, but then how do we sift through that complex data, as you guys are very familiar with on the biological side of things, and be able to tell out what these chemicals are. So same game that we play in biology, environmental chemistry. So we can kind of develop our tools that we're developing both at Yale and with Innovative Omics for any kind of application. So a lot of these tools spread over, which is exciting. Actually, this whole framework was developed for lipids, and then we kind of taken it and advanced it for this application. So our workflow is usually we have extraction blanks, pools, and samples, and we use those pools to get deep MSMS. This works better in biology where blood is blood and you might expect different levels of metabolites, lipids, proteins, but you don't expect that many completely new or chemicals from one group to another. I mean, if you do find those, that's fabulous, but generally it's just different levels. So you can use these pools and get deep MSMS. A little harder in environmental, where each environmental sample might have completely different chemicals. Then after you acquire this deep MSMS and full scan or some kind of data dependent analysis on all your samples, where you can get full scan data out and your extraction blanks, literally all you do for this software, both for the lipids and PFAS, as well as now we're going to polymers, is you drag those files on and click go. That's literally, and it will read the extension .b.raw. With whatever it is, and it will optimize the parameters accordingly, or use optimized parameters. So then it does file conversion, peak picking, blank filtering, and annotation and grouping by series using these kind of mass defect plots. So all your data is organized by series. If it was lipids, it could be classes, et cetera, et cetera. Then we also developed community-based libraries so that you can set up your own rules. Where's the repeating units within your structure? What kind of fragment rules? What kind of neutral losses? And I'll generate all those masses in a format that can be used by the software. You can also use your own peak picking and post-processing. So this is an example from Agilent, or if you're Thermo, you could do Compound Discover, or you could use MZMine XCMS. You know, peak picking is very challenging, as many of you for non-target analysis probably know and have tackled and continue to tackle. And so if you have your own optimized parameters, probably better use those and then just use this modular version to annotate those feature tables. And then it gives some kind of Szymanski related score to those annotations. So this is the game that we all play of how do we narrow down, like a guess who game, what our chemical of interest is. Oops. So you could say, does it have rosy cheeks, pulls out, beige dress, darker tint, until we get to that one chemical. So what are all those pieces of evidence we incorporate in these software suites for all our different applications? So one is to find homologous series with retention time and exact mass. These are very helpful for organizing your data. I'll show you how we do that. And then of course there's exact mass library hits, mass defect filtering, which is useful in PFAS, where fluorines have very unique mass defects, negative mass defect generally. So you can use all these filters. Exact mass alone is very tentative, but can help you narrow down what you're looking for. But these homologous series are very nice. Even if you don't have MSMS data, they can give you some form of confidence. So in this case, we're normalizing to CF2, be CH2 if you're looking at lipids, kind of different CF2O, there's different types of PFAS, so ethers. And they should form these straight lines because now CF2, we decide that its mass is 50.00000 and everything's normalized to that. And so anything that just differs in the number of CF2s in their chains, which is generally how PFAS work, will be grouped as a single series, and they should also fall in intervals of 50 Daltons for those CF2s. And so we can pull out classes, you could say, or series of PFAS just using this algorithm. The next, so this is an example from some AFFF data, so firefighting foams. This was acquired by Carrie McNano, who worked also very hard on validating these annotations and doing her non-target analysis by hand, which we then compared across since she had already published on that. We got 95% true positives or 5% false positive rate for using some kind of class-based fragmentation rules. But here you can see all the different series that form and the different classes that you can detect in AFFF just using the software. How useful it is to group them this way. Along with homologous series, again, or expand this to your metabolite lipid applications, especially lipids are things that form groups. Metabolites are a little harder because they're each one-offs. You should see, along with that space and mass and the right mass defect, you should also see a very clear pattern in mass charge versus retention time. And I actually find that this is extremely valuable for pulling out false positives. You can see one in this plot already on the far left, and giving more confidence to those that fall within these trends. Most things don't fall within these trends if they don't belong to the same homologous series. They don't form these perfect trends. And so very important for distinguishing whether these compounds are real. So here's an example. Pull out a series, it had the right masses and stuff, but if you mention time mass plot, no pattern there. Pattern, no pattern. So you get the idea there. Another way is this class-based MSMS, where we take standards, very common in lipids. You know, the PC has the 184, things like that. So I'm using lipids since you guys do a little more biology, but maybe also not a familiar reference point. But in PFAS, also you have these classes that have specific fragmentation, whether it's the sulfonic acid, carboxylic acid. And so, you know, there's probably 136 subcategories. This was a while back, so probably more now that have been defined. And so we try to find standards for these and do this kind of process where we decide where the repeating unit is. We run the standards, figure out what some of the most common fragments are. You can see on the bottom, C8, C10, C12, and C14 fragmentation. And then we pick some of those top abundant fragments or distinguishing fragments and use those as rules to species. And again, that gave like a 5% false positive rate. So it works quite well, even though we're not using intensity at all. It's just, do you see these masses? Do you see this exact mass of the precursor? Do you fall within homologous series? Those kind of things. This is some data from dried blood spots. I think this is really interesting data. My one caveat is we didn't collect the blood ourselves, although we did run blank blood spots. So we don't have a perfect blank. So depending on what syringes they use, how they stored it. So we need to look into that. But if that isn't a major contributor of PFAS, we've just discovered like 50 new PFAS that have been looked at blood, which is a big deal in these dried blood spots. So this is exciting. And this is just showing how MSMS can find different isomers, etc. So other things we can do is this fragment screening, where we take some of those common fragments, or 777 fragments in this case, pick a nice number, that we got from standards, and screen the entire MSMS data for these fragments. We can also predict using kind of manually defined by an undergrad we had, looking at the structure of the molecule, looking at the fragment, and trying to decide which substructure that fragment came from. Some are easy. Pentafluorosulfide would be SF5- fragment, easy to link. Some are not so easy. And so we took the ones that we felt fairly confident about their links. And then we're able to predict fragments by taking the entire EPA database of these molecules, looking for substructures and assigning fragments that are related to those substructures. Any questions so far? I talked a lot, so.
Can I ask a question real fast? So this is
Yeah.
really fascinating stuff. One of the things that I'm interested in is, you know, all the time you hear about how we get all these big benefits from things like ion mobility. And using something like that in this case, when retention time is such a clear differentiator, is that something that will help a lot? Does the complexity warrant that, or is it overkill at this point?
Yeah, I think, all right, so this is my opinion, and I know a lot of people have strong opinions. So from me using ion mobility, both in this application and lipids and metabolites, I think it's really good in a targeted area where you can have especially different sodium adducts and certain things that really resolve and form these kind of clusters that can be resolved that you can't resolve in retention time. Generally, as we know, the resolving power of ion mobility is less than retention time, and it's generally not orthogonal is what I find. Generally, if you can't separate in retention time, you can't separate in ion mobility, and then there's a lot of exceptions which people publish on. But it does give some level. I think what's really useful for ion mobility is the collision cross sections, because predicting retention times is extremely difficult. Calculating cross sections is relatively easy. And so as far as adding confidence to these identifications by having libraries or predicted CCS by class or something, it doesn't have to be like predicted universally given any structure, would add a lot of confidence to the annotation. So I think it will add confidence, but not better resolving power in many ways. But we haven't, I know Erin Baker has done a lot of work on separating PFAS. So she'll be at CTAC, NTA, and probably SMS too. So I'll be looking forward to seeing what she found out. And I'm sure there's definitely ways. And then there's SLIM too, right? So that takes the resolving power and increases it dramatically. So that might help.
Cool, thank you.
That's just my thought. Let's see. Numerous FluoroMatch. Oh yeah, so a lot of collaborators. We're working closely with the EPA, especially in Rhode Island, the group there, led by Mark Cantwell. 3M expressed interest. We haven't, they keep, things take a while, but that would be very exciting given that they are the producers of this, one of them. Cyclopure is like a water filtering company for PFAS. So just exciting to be able to, by leading these trainings, develop tools. I am doing some applications on my own, but I get to empower other people to do their applications and get to see a wide range of scientists and their cultures in their lab, which is a lot of fun to do that. And then get to see some of these cool applications like leachate is really good for getting the average of all the stuff we produce and use, you know, goes to the landfill and then kind of gets extracted out by this acidic, intense environment. Some of it will be transformed too. And then you can take that juice and try to just understand what's out there. And we found 300 plus PFAS in these. So these are things that are out there in consumer products that we're measuring in the average garbage juice. Cosmetics, we're looking and we're doing a big study now. So that will be interesting. They use a lot of PFAS in cosmetics, which is not great given the fact that it's getting absorbed for sure. Definitely probably a major exposure route that we haven't looked into much. AFFs and obviously the industrial influence as major sources. And then we're also looking at the human body as well as mouse models. So this dry blood spots I talked about, we Vietnamese populations look into. We have these wristbands that are really interesting. And we're doing more large scale exposomics using GC-HRMS, looking for pretty much any chemical on the NIST wide range libraries, which is already set, which is super nice. But we're also applying this to PFAS and generating our own GC libraries. So if you walk around, it has a little PDMS bar and it absorbs things from the air, both particulate matter, as well as any volatile or semi-volatile chemicals that are on their own. And then we get a time average, let's say over a day, usually or 2 days, maybe sometimes a few hours if you're in a very contaminated site. And then you ship those to the do some direct analysis via GC, and we can see whether you're walking around and getting exposed to PFAS in the air. Same thing, we've done this for COVID, as well as other environmental chemicals, plant-based volatiles, etc. Yeah, I did a lot of different designs that Elizabeth on the far right loves to develop all these different designs which can answer different questions. So here's a EIC and GC. It's really nice because you have this hard ionization and you get very significant push of all that fragmentation to the low mass range. So you can look at CF3, C2F5, some of those low mass fragments that are universal for anything that has a carbon fluorine chain and just see what's out there. And so we can see that in the shoes, especially, which is going to collect more facing up, more dust, we can see a lot of PFAS, but even in the chest and the wrist, we can see some. So kind of validated that we can at least see stuff as people walk around and start correlating this to their environment. Find out what our PFAS exposures in the air are, which no one's really looked too much into. Here's some more blood spot data. Just giving you an idea of why the software is important. This is what you first see. I'm gonna actually pull up the actual software. Feel free to ask questions. Can you see my window again?
Looks good. Yep.
Okay. So this is the software, and this is what we're going to design for lipidomics and other analyses as well, polymers, polysorbates, things that are used as additives in vaccines, et cetera. And it's really nice. So it's this interactive platform after we process it through our whole data analysis workflow that gives you a visual. And you can kind of scroll over, see information. Okay, so this is a sulfonic acid. It's got a score, a mass. You can click on anything. See its MSMS profile annotated. Okay, one of the majors, C2F5O, usually an ether-related fragment, but also maybe related to this oxygen and the sulfonic acid. You can, we can refresh by clicking anywhere. You can screen by fragments. So I know C3F7 is a common fragment. Okay, here's a series of PFAS. Okay, it looks like carboxylic acid is a common one. So you can do a lot here. I won't obviously do a whole tutorial of the software, but just kind of give you an idea of how easy it is to use and kind of the power of which it gives you. You can add all kinds of graphs and things that will be interactive with this data. So we'll add statistics and that nature. Let's share back to the PowerPoint. I'm going to try to give a little more time for discussion. So it's not just you listening to talk. So this is Emily Perry from Agilent, ran this data on a 6546. Elizabeth did a lot of this extraction and sample prep. And you can see some of this. This is dry blood spots, unique chemicals. And I'm not sure, you know, the second from the top is a kind of Teflon related chemical. It's just a perfluorocarbon chain, maybe with an H substitution. We're not sure the ionization product of that. Some phosphates, which are related to food packaging, ether lipids, which are really increasing now relative to the past, and then your traditional sulfonics and PFCAs. So a lot of interesting chemicals here that I'm not sure have been, I know many of these haven't been found in blood before. So again, we have to look into that whole process, make sure it didn't come from a syringe or anything. But exciting work there. And here are some of the major leaders to this project. Again, we do trainings at Innovative Omics. So if you guys need lipidomics trainings or something like that, I can do a site visit, things of that nature. So I have a few slides on lipidomics to show you that, or we could end here and discuss. I don't know what people want to do. I'll just race through these slides. Lipids are important. Lots of good biology there, and a lot of bioactive lipids. So if there's a small change in lipid, there's a large phenotypic response. And so these are good drug targets as well as for biomarking, creating diagnostic tools, things of that nature. And they don't organisms. So unlike proteins, you don't have different isoforms. So if you do an animal model and you find a certain lipid has changed, that lipid will probably be the same in the human, especially in mice. We share most of the lipidome. The mechanisms in biology might be slightly different. Chemical itself, it's not going to change from generally. And one of the big applications, so we've developed, 500,000 species predicted spectra from standards, et cetera. But one of the very interesting ones that I've been looking into that's kind of novel and later in the last few years I've been doing is these oxidized lipids, which are kind of universal indicator stress. So this kind of links the PFAS and different environmental exposures to the biology. Can we do these widespread exposomics where we take what we're exposed to and relate it to our health, given the fact that most diseases are probably related more to our environment and diet and things of that nature than to genetics. So here's the oxidative damage. These allele hydrogens on these lipids make them very prone to oxidation. So they oxidize easily, and they're generally some of the first biomolecules to oxidize when they're stressed in the organism. They have lots of isomers, multiple oxidations. You can get in long chain, hydroxy, hydroperoxy, ketones, so on and so forth. And then all the different possessions on the glycerol backbone. So from a computational standpoint, both a headache and kind of exciting. We can produce over 1 million triglycerides. How many actually exist in nature? So looking at a very high level analysis where we looked at these oxidized oils when you fry it or bake it, and we were able to find 3,000 oxidized triglycerides using the software. So quite a lot out there. That's just features, or actually, no, I think that was the number, but there's probably a lot more out there. We had to limit our libraries just for computational time. Kind of the take home here is olive oil, much better than walnut oil. Walnut oil goes rancid. Soybean oil is pretty good too. Baking or anything where you have a large surface ratio. So anything where there's a lot of air exposure and you put a little oil on your potato and you put it in the oven, that's actually going to have a lot more oxidation than something that you're frying in a pan, deep frying, where the air doesn't have as much contact, is what we kind of found out. And the types of oxidized lipids change too. We've also looked at alcohol and liver disease in a mouse model. This was done with people from Yale, George Capardi, and Vasilis Vasilis, the PI on this. So increasing ethanol over time. Histology showed barely anything, a little bit of steatosis, so some fat buildup. Not much else. But in the lipidome, almost the entire lipidome shifted dramatically in these liver tissues, and in the blood there were some significant ones. And the oxidized lipids took the cake. So earlier on than any other lipid changes, we saw these oxidized lipids in the liver. So really interesting. Right after 2 weeks at a relatively low level of alcohol, we're able to see the spike, and then orders of magnitude increase in oxidized lipids. This same one in liver was also found in plasma and also found to increase. We didn't find many trends that were the same between liver and plasma to the exact molecule, but this was one that, while the trend's not exactly the same, they both increased. So potential marker. Obviously, the issue with oxidized lipids is any kind of stress might produce, but we are finding that different stresses produce different oxidized lipids, and I don't understand that mechanism exactly yet. Yeah, so I'll close soon. I just wanted to share something more about lipids that I find very fascinating, is that you can have a complete shift in the lipid membrane or in lipid pathways to use completely different molecules when affected by different environmental perturbations. So for example, here we have seagrass in low phosphorus. It loses these lipids in its membrane that have phosphate in them. So your phosphatidylcholine, phosphatidylaceric acids, phosphatidylethylamine, all those almost disappear. They decrease dramatically. And then orders of magnitude increase in these other lipids that you can see their structures. And if you line up just the first two, PC to DGTS, you can see how structurally similar they are without having to use the phosphorus. So completely re-engineering, and all these plants can do this. Many plants can do this. Re-engineering their membrane to not use phosphorus and use a totally different chemical to create a different membrane. And then obviously they look different physiologically. And this isn't the only case where this has been shown. In bacteria and certain things that evolve faster, they show that they change their membrane composition of lipids when there's plasticizers in the environment, and those plastics get incorporated in the environment. So they want to keep that membrane fluidity. So I just think it's fascinating with this lipids kind of take-home message of how fast they can change and how dramatic they can change with different responses to the environment. Why I think they're so important to study from a disease and a health standpoint. And that is it. I'm gonna stop sharing and we can open up for questions if we have time. It is 5:40.
That was really awesome, Jeremy. I have a quick question on the looking for PFAS in blood and things like that. Is there? I'm not super familiar with this area of research, but I think I had heard that there's really no biotransformation that goes on, or is there like some known biotransformation? Like, are you able to kind of apply the known typical drug biotransformation mechanisms to your list of like PFAS targets to then generate a whole other list of potential targets to pull through the FluoroMatch software?
That's a really good question, and definitely something that we're working on. I'm actually supposed to present that CTAC in a week. I haven't gotten the data yet, but we've used Biotransformer to kind of just plug in these pictures, because if they have a carboxylic acid or sulfonic acid, we do show different kind of functional groups, conjugation, different amino acids, things of that nature.
Mm-hmm.
And so, yes, they do form some of these biotransformation products, especially additions. It's hard to integrate that chain, but along the functional group, they can play all similar chemistries and some different ones. So yeah, it gets astronomical, but even, this is really interesting, those carbon fluorine chains look like a fatty acid, right? So the question is, do they get incorporated into lipids? And people have shown with a few carbon fluorides and rest hydrogen that they do get incorporated into lipids. And now the question is for fully fluorinated chains. I think people are starting to show that we now have lipids that have carbon fluorine chains, which is interesting. Never before found in nature.
Yeah, you have some like hybrid, like cyborg lipid things.
Yeah. We're all partially 3M inside.
Oh man.
Yeah. See a hand, Joe.
I got a yeah, Joe, go ahead. Please ask a question. I had some questions for Jeremy, but
I just have one quick one. And, one of it comes down to, I'm in the same boat as Gene. I don't know a whole lot about PFAS or how they work, but, looking at the structures, it's basically Teflon, right? So this is fluorous, and this is known to have an extremely tight interaction with protein binding in general.
Oh, interesting. Yeah.
Is this something that you guys encounter when you look at things like half-life or circulating, residence times in plasma, that you just see that it sticks around forever and forever?
Yeah, no, it's an interesting concept. So women who menstruate, or I imagine people who donate more blood, I've been thinking to donate more blood, probably get rid of it more than those other people who don't, because then, they're actually losing some of that blood. And they've shown it for the menstruation. Yeah, they have shown that albumin levels. Interestingly enough, we did a study on these fish that had a disease, and we wanted to see if it was PFAS related. And it was very hard to tell because the disease affected the protein levels in blood, and those that had very low protein levels in blood had very low PFAS. Those that had very high protein in blood, very high PFAS, just probably based on.
So there's no clear elimination mechanism. I guess if biotransformation isn't happening on things that aren't, amenable to it, it just kind of stays around.
Yeah, I think it has like a 50-year half-life. Don't quote me on this. And
Okay.
yeah, so it's quite a long time. And rats, for some reason, I have to look back into it, have a much lower half-life, so they're able to get rid of it a little easier. I haven't looked into the mechanisms.
That's interesting. Thank you.
Makes mouse models hard. Harsh.
Yeah, I have a question. Thanks a lot, Jeremy. Wonderful talk. Again, this is a new topic to me, PFAS. I don't know much about it, but from listening to some talks and your talk, I was wondering whether, there's environmental exposure and like biological systems and proteins, modifications, biotransformation. But has anybody looked into very large scale, like proteomic scale analysis of what proteins are mostly susceptible to these modifications, or if they're getting modified, if covalently, or I don't know the mechanism, but is there, can you find these reacting with proteins, certain functional groups, or is that not happening? I'm not quite clear about how these might impact proteins themselves. And
yeah, no, that's a good question, and I don't know the answer. Is my answer, and then I do know that, or at least I always imagined, and I'd have to look in that they're just adhered via they're both hydrophilic and I mean hydrophobic and not and lipophobic. But yeah, I don't know that answer.
I believe there's some kind of free radical mechanisms, right? I mean, you can have proteins that kind of pretty much can be affected by some kind of free radical mechanisms, right? A lot of the KRAS proteins are driven by free radical mechanisms. Is there some way we can generate something in that nature for reactivity with proteins and altered structure and function? I'm not sure whether people focus on those aspects, or whether it's more on the major state of proteins that your people are mostly interested.
Yeah, I don't know. What do you think the impacts would be if it did have these chemistries with the protein?
I have no clue. I mean, I'm wondering whether there's some research areas on this to look at these in more detail.
Yeah, I mean, I'm just Googling and I don't see anything on protein efficiency.
So I guess the answer is no. You're the expert, so I guess I just a naive question that I had is that important, and have people looked at these in more detail across the entire proteome? You alluded to earlier that it's just integrating with other lipid molecules, similar structures. Well, if that's happening, maybe it is impacting other areas of biology that we're not aware of.
Yeah, no, that would be really interesting to look at. I think this article is not very interesting, but it says albumin is the major, but duh, because albumin is just a dominant protein. So yeah, I don't know, and I think that would be interesting. I don't think a lot's been done at that mechanistic level. A lot's been done with the PPAR pathway and the peroxisome, and some of those. And then in terms of gene expression and protein expression, a lot's been done on actual. I mean, it's challenging, right? How do you characterize structurally what is happening at that level? People have looked at like fluidity changes around organs. Because they have very unique fluid properties, and they sometimes concentrate around these between organs and different cell membranes and things of that nature. And so that definitely has an effect, alveoli and things of that nature. But I don't know about proteins specifically, how they affect the structure and function.
Thank you.
Yeah.
Any other questions from the audience? typed it in, I could read it for you if you're too shy to ask.
Just, yeah, I have a question. Just for curiosity, I mean, the NIST is building databases, right? Libraries and so forth. Are these spectra that you're collecting, are they going into some kind of repository, or is it just for your local consumption? Are you?
We should be, right?
Yeah.
For the GC high resolution data, we are compiling it in a way that we hope to distribute it as MSP files, or we know we will. I mean, that's going to kind of be the product of it as MSP files, and we can post it wherever, mass bank, or they're going to be somewhat predicted. For all the PFAS spectra, we have the spectra, we have not. I mean, it's a little work to put it in the right format, do any kind of annotation. So we haven't done that with the GC Orbi data. Although all the libraries, everything's open source, the list of masses, fragments, precursors, but you know, it's not an MSP file or something open source necessarily. I mean, it's open source, but not something widely used. So for the GC, yes. For the LC, we have not done that.
There's a question for you. It is, how are oxidized lipids identified? And the question's from Rena.
Rena. So you get some interesting fragmentation. You get, for example, from a hydroperoxy addition, so OOH on a fatty acid, you can get a M-H2O2 type fragment, I believe. I have all of those MSMS spectra I can send you, annotated, and there's an article on it. Then for like a hydroxy addition, you might get M-H2O. You could get M-2H2Os for 2 hydroxy addition. So you can kind of get these border losses that don't normally occur for most of the lipids, border losses, but that don't normally occur, and that gives you some evidence. And then the great thing with lipids is the ester linkage, where the fatty acid links to the backbone, is the major fragment area. And so if you have an oxygen attached to a fatty acid, you'll know, because the mass of that fatty acid will be that plus an oxygen. If you have a high enough resolution instrument, you can even look at the isotopic profile if you have a wider isolation window. We don't do that. We just look at the mass of that fragment. So those kind of, and then you can get water losses and things off the mass of that fragment, showing you, oh, this is a hydroperoxide versus a hydroxy versus a ketone. So those kind of things. Generally, you just know the number of oxygens and which fatty acid they are on. You don't know the position of that oxidation generally, and you don't know necessarily the type of oxidation, although you can have certain distinguishing. You might not know whether there's 2 hydroxys or 1 hydroperoxy. So in-depth answer to that question. One other additional thing I think that's really interesting is all ions. So if you do all ions or all ion fragmentation, as Thermo calls it, all ions, I think it's Echelon calls it, either way, or some kind of swap approach on all these kind of approaches, you can pull out for fatty acids, well, PFAS, but fatty acid fragments, and show, if I know what the mass of 18:3OH is, I can look across my entire chromatogram and see which lipid regions, how many lipids, a rough estimate, have these fatty acids. And so I can see if it's a universal fatty acid or just something that's found in one species. So that's kind of a fun way to do it too. You're welcome. Yeah, feel free to email me any questions related to any of these topics. Throw it out there, and then hopefully I'll get to you.
Thank you. So we'll give it a few minutes for any more questions to bubble up. Maybe not a full couple minutes of silence, but yeah. But it was really awesome, Jeremy. It's really the interface of the software is very pretty. So it's great to see
Yeah.
powerful software that actually is nice looking as well.
Yeah, I mean, until a year ago, and I've been working on this for more than 9 years, like, or I think it's been 5, feels like 9 years. It's been just an Excel sheet. You get out a CSV, and then you have to look through it. And I realized how archaic that was now that we have this interface and how many things we were missing and how many false positives. Visualization is just essential for kind of validation.
Sure. All right. Well, I guess we're going to wrap up. No more questions. Thank you so much, Jeremy. Harsha, you want to say any last words or any other?
Not really. I think you're the newly elected chair, right? So I guess so is all yours, and you're in charge. Thanks again, Jeremy. Wonderful talk. Really appreciate you taking the time to come and give us this wonderful talk. Thank you.
Thank you for inviting me.
All right. Thank you, everybody.
Bye.
See you at ASMS.
See ya.